The concept of evolution is proving to be less random than what was previously believed. Greetings and salutations to everyone. In today's video, we will be delving into a study conducted by the University of Nottingham that sheds new light on the predictability of evolution. Contrary to previous beliefs, this study reveals that evolution may not be as unpredictable as once thought. This newfound understanding has significant implications for tackling real-world issues such as antibiotic resistance, disease, and climate change. By exploring the influence of a genome's evolutionary history, scientists can now identify potentially useful genes. This groundbreaking research challenges long-standing assumptions and opens the door to innovative genome design, climate change mitigation, and medical advancements. So, without further delay, let's dive into our discussion. Scientists may now have the opportunity to investigate the potential usefulness of certain genes in addressing real-world problems like antibiotic resistance, disease, and climate change, thanks to a recent groundbreaking study that has revealed evolution to be less unpredictable than previously believed. Published in the Proceedings of the National Academy of Sciences, PNAS, a groundbreaking study challenges the widely accepted notion of evolution's unpredictability. It reveals that a genome's evolutionary path can be shaped by its own history, rather than being solely determined by various factors and chance events. Professor James McInerney, along with Dr. Alan Bevan from the School of Life Sciences at the University of Nottingham and Dr. Maria Rosa Domingo Sananes from Nottingham Trent University, spearheaded the research study. Professor McInerney, the study's lead author, expressed the groundbreaking nature of this research. He stated that the implications are truly revolutionary, as they challenge the previous belief in the randomness of evolution. This newfound understanding paves the way for a multitude of opportunities in synthetic biology, medicine, and environmental science. To address the question of whether evolution is predictable, or if the evolutionary trajectories of genomes are influenced by their past and therefore not predictable in the present, the team conducted an analysis of the pangenome, which encompasses the entire set of genes within a specific species. To address the question at hand, the team dedicated several 100,000 hours of computer processing, utilizing a machine learning technique called random forest, in conjunction with a dataset consisting of 2,500 comprehensive genomes from a singular bacterial species. Once the data was inputted into their advanced computer system, the team proceeded to create gene families for each gene within every genome. Dr. Domingo Sananes explained that by utilizing this approach, it would be possible to make meaningful comparisons between genomes that are similar in nature. After identifying the families, the team proceeded to analyze the presence or absence of these families in various genomes, examining the patterns that emerged. Upon our investigation, we discovered instances where certain gene families were absent from a genome when a specific other gene family was already present, while in other cases the presence of certain genes heavily relied on the existence of a different gene family. The researchers made a significant finding: an unseen ecosystem where genes have the ability to either collaborate or clash with each other. Dr. Domingo Sananes emphasizes that the interactions between genes contribute to the predictability of certain aspects of evolution, and furthermore, we now possess a tool that enables us to make accurate predictions. According to Dr. Bevan, this research allows us to delve into the genes that provide support to antibiotic resistance genes. By understanding these supporting genes, we can develop strategies to not only target the main gene, but also its auxiliary counterparts in our efforts to eradicate antibiotic resistance. By employing this methodology, we have the ability to create novel genetic structures that have the potential to advance the development of pharmaceuticals and immunizations. The newfound knowledge we possess has paved the way for a multitude of groundbreaking revelations. The potential impact of the research is extensive and has the capacity to result in significant outcomes. With the advent of novel genome design, scientists now have the ability to design synthetic genomes, offering a clear path for the deliberate manipulation of genetic material. In the fight against antibiotic resistance, Gaining an understanding of the interconnectedness of genes can play a crucial role in identifying the ensemble of genes that enable antibiotic resistance, thus opening doors to targeted treatment approaches. The findings from this study have the potential to provide valuable information for the development of genetically modified microorganisms that can effectively capture carbon and break down pollutants, thus playing a significant role in combating climate change. The potential for gene interactions to be predictable has the capacity to greatly transform personalized medicine, offering novel measures for assessing disease risk and treatment effectiveness. To support our channel's growth and ensure broader awareness, kindly hit the like and subscribe buttons. This will help us reach more individuals and disseminate valuable information. Thank you in advance.